Hello students, so let us continue our discussion in this biomolecular spectroscopy and especially about how we can use these techniques in pharma and biopharma applications, right. So, we wanted to just today in this lecture we want to emphasize application of Raman spectroscopy in pharma and biopharma industry. Right. Raman, as we have seen, it is a very important based on the based on similar principle like IR vibrational spectroscopy, but it is just a low sensitive in terms of the intensity outcome comes inelastic scattering that we have gives you, and you require a laser source that we have already discussed during our classes. So, where we can use the Raman spectroscopy in pharma and biopharma, that is what we will be discussing today. As we know, right. So, Raman spectroscopy has a global scientific legacy. Let us revisit this legacy. So, and why it is becoming important? Because it has entered into translational pipeline. Earlier most like a discovery phase where scientists were using Raman spectroscopy, but now with advancement of the detection technology, the laser technology and all those, it has turned into a multi-billion dollar industry. If you look at some of this market explosion today, as of today, like last year it was 3.66 billion US dollar, and it is expected to grow in one decade up to 20 billion dollar. And if you look at this is going to with CAGR of 20.42, that is a dramatic increase that we are going to see the usage of Raman spectroscopy in this in the field of pharma and biopharma. So, this is going to be playing a phenomenal role in explosion of the application of Raman spectroscopy. So, it started say a decade ago from lab to field. How and who? As we know, Raman spectroscopy says C.V. Raman is the pioneer. He was working in cultivation of science in Kolkata 1928. He discovered, but now it is a power handheld device, can go to like any place, field, clinics, everywhere worldwide this is used. And size revolution, like it has reduced 90% smaller than original size. So, it is a very like a handy, less than 4 kg, and class 1 laser safe it is. So, laser has reduced, the instrument has reduced and it has become portable, and because of all those technological advancement that we have, we are seeing that application is going day by day. So, we can deploy everywhere. In the teaching lab, even undergraduate students can do, and I am going to show you where all we can use it. So, undergraduate lab can do, you can have even at a custom checkpoints, right. So, lots of like goods material are transported across the border, and custom officer can use the Raman spectroscopy for checking it. You can have even in the remote setting of the clinics, somewhere in the remote village you can take this and do the detection. Some of those example also we will be looking at. And even you can deploy in the field as a field operation, we can use Raman spectroscopy. So, now technological evolution that has happened, what has happened, laser got integrated and it is computerized, and with the now boom of AI, everything is coupled with AI and miniaturization happened, and that is why now it is up to 4 kg and you can take it from lab to the field. That is the advancement we have seen in Raman, right. So, this is Professor C.V. Raman, initiated his research in cultivation of science, that is just a schematic that is shown, and here he was using this that was discovery by Sir C.V. Raman He in 1928 he got the molecular light scattering that we had already discussed, but you know technology and science goes hand in hand. It was like he showed conceptually, he got Nobel Prize in 1930, but you see the technology helped it in propagating. So, in 1960 when laser was integrated with the Raman, and from 60 to 80 when laser was propagating, Raman also got boom, and now with computer digitization and processing, the data acquisition and decoding becomes very important. And now with AI everything becoming fast, data interpretation, data handling becomes, so big data can be analyzed in seconds, and that is what made this CAGR grow with 20.42. So, that is what $20 billion industry it is going to have. Now, as just for an schematic I can show you this, these guys are using this handheld Raman device to look at what has been like if some narcotics or something is coming, they can identify using molecular fingerprint. So, like old Raman was like that, a bigger one, now new Raman instrument is like this. It is a handheld device, doctors can use in clinic, custom officer can use in at airport or at the port, they can use it, and that is propagating it. So, since we were discussing pharmaceutical and biopharmaceutical, the identifying in counterfeit drugs for ensuring the 100% medication purity is one of the important part, and here Raman plays an important role. So, if you look at counterfeit crisis, actually there are people who are doing all this nonsense, and because of that the quality and efficacy of drugs goes down, and somebody has to really like see if this is the drugs are safer and they are not adulterated. So, market was about 410 million, and that is going to go in another 5-6 years, double, more than double, actually 980 millions by 2031. It is a fighting 200 plus billion global drug fraud, and this fraud you never know as a consumer that what is good and what is bad, but somebody has to catch it, and here Raman plays an important role. Because of this, actually you can save the lives. So, lives saved at the border in 2024. The Nigerian custom officer used the handheld Raman devices that I showed you here, these devices, and basically stop the shipment of anti-malarial, they contained chalk and yellow dye. You see, they are importing, Nigeria was importing actually anti-malarial drug, but these people are selling to Africa, to Nigeria like a chalk and yellow dye. That is a crime, but some like it is just the human ethics that is doing it, but somebody has to really catch it, and Nigerian custom officer did it, and he saved like with 60 seconds of his scan, he saved life of hundreds of children. In our country, Delhi hospital story, Dr. Sharma received insulin for diabetic patient, right. This to treat a chronic disease such as diabetes. The Raman spectroscopy revealed the 0 active ingredient. So, these guys sold the insulin without actually being insulin inside, that is nonsense, right. So, but that was done, and Dr. Sharma actually could identify it. So, what appeared the legitimate would have caused the fatal hyperglycemia in dozens. Similarly, 99.7% accuracy, transmission Raman can detect the fake drugs through unopened packaging, no breaking of seal, no destroying of evidence, you can catch all of these. And then the fentanyl deadly games, SARS, surface enhanced Raman spectroscopy technology identify the lethal analog in picogram concentration. In Vancouver, this caught the counterfeit Xanzase pills containing enough fentanyl to kill about 50 people per pill. So, that was caught by SCRS. And FDA and WHO trust, like when regulatory agency worldwide take a stake of public health on technology, you know these guys work and they use Raman spectroscopy. Right. So, where we use Raman spectroscopy in pharma. So, Raman is becoming one of the most prominent analytical tool for pharmaceutical application, all the way from validation of raw material. Your raw material has to be very, very crucial. If the raw material is not correct, your drug product is not going to be correct. So, quality assurance or quality control starts early on, and that is from raw material, and Raman plays an important role. It is a crucial role in pharmaceutical industry by addressing the key needs such as drug purity, authenticity, efficacy. Indeed, this technique is used to verify raw material, detect counterfeit drug, monitor polymorphic form, and analyze the drug formulation that are there. So, here again Raman has role to play in each of these, the counterfeit drugs, the polymorphic form, and probably I am going to show you some of those examples. So, good part that it requires minimal sample preparation, whether it is in solid or liquid, you can do it, provide the precise molecular identification and characterization by offering real time inline monitoring with a detail of a spatial distribution information. Actually, Raman enhances the quality control. It not only quality control, it also supports the regulatory compliance. This is extremely important in the pharma industry, regulatory compliance. And if we start checking the raw material or detect counterfeit or monitor the polymorphic form, if we know that correctly, we can accelerate the drug development and effectively meets the industry challenge for mitigation, and we meet the high standard, and also we safeguard the public health, right. So, this is some of those usage, like at the FDA anti-malarial drug that I was talking to you. You can find it, the authentic drug signal is like 1, and you see this is the chalk and yellow dye is like this, and you can look at the intensity that is there. So, in 60 seconds 99.7 accuracy you can find, and you can save the life of so many children that could be done, right. So, application in modern medicine, one of the Dr. Cheng's story I will just tell you what he did. He is from Chen, he was from Shanghai, and he is a neurosurgeon. He removes the glioblastoma tumor. Before actually Raman, he guessed that cancer ended, but now by looking at the Raman spectrum, he knows that reoccurrence rate dropped only from like 38 to 7%, it is not completely gone. So, he got it 93% sensitive, 91% specificity in real time, less than 1 second. Now, the other story I just want to tell you, the Maya's second chance. She is 34-year-old breast cancer patient, Maya. She underwent with lumpectomy, and Raman proves that Raman could detect basically microscopic cancer that surgeon could not see. Now, complete removal was on first surgery, no reoperation needed, no trauma, no delay in treatment, and her disease could be diagnosed and treated properly. The ACRS nanoparticle, actually 100-fold signal enhancement you can do. So, here you can just probe the tumor correctly like a heat seeing missiles, and then you can illuminate that tumor region, and then doctors can precisely do the operation. Another example of Raman in modern medicine is Boston Children's Hospital innovation. The pediatric brain surgeon uses Raman to preserve the developing neuron tissue and removing the tumor, that reduces positivity margin from 40 to 5%. You can also do the fatal early detection. So, blood based Raman screening caught the Mrs. Rodriguez cervical cancer 2 years later, like 2 years before. Traditional pap smear would have done it. So, that was with 95% accuracy, and then intervention can start much before disease appears. So, all of these, even in modern medicine, Raman is going to play an important role. Coming back to the pharma story, right. So, say a tablet is made up from the it has mainly two things. One is called API, active pharmaceutical ingredient. other is excipients or adjuvant or whatever it is called. So, but it is a small chemical molecule, API is a small chemical molecule. There is a possibility that it has a different polymorphic nature, right. It can be crystallized in different polymorphic nature. So, polymorphism is a big headache for pharma industry, and that actually can change the solubility, stability, bioavailability, many other things, and even efficacy. So, Raman can actually detect the polymorphic form of API, and it ensures that right polymorphic form is formulated and drug performance can be ensured, the safety can be ensured, shelf life can be ensured. So, how do we do that? Identify and control a specific polymorphic form, like in a pharmaceutical product, ensures the consistent performance, efficacy, regulatory compliance, and ultimately protects the patient health by enhancing that whatever drug is there is correct one, it has a maximum therapeutic effectiveness, and one can ensure the quality of these drugs. So, in discovery and development, Raman spectroscopy can be used. Differentiate between polymorph by analyzing their unique Raman spectra, quantify the property of different polymorph, right, in a mixture by analyzing intensity of Raman peaks, that we show that if the peaks are different, you can see two polymorphic forms, their intensity can tell you something about that it is two polymorphic form with a different composition and concentration. It can also predict the shelf life or stability of the polymorph. If the intensity goes down, you know that the one polymorphic form is getting degraded. So, Raman spectra sample can be treated in different chemical environment with a temperature, with humidity, and all those stress associated tests you can do. So, here as an example for understanding the polymorphic nature of API. Now, you can see here are two forms that are there for a API, form 3 and form 1, and you can clearly see that spectra, this is the Raman shift in centimeter inverse, you can clearly see some of these peaks are different and their positions are different, right. So, Raman could differentiate the form A, which is in blue, and form 3, which is in red, and it can actually diagnose or detect the peaks which are here different, right. So, diagnostic peak at say 750 centimeter inverse versus 720 or whatever. So, here again I can clearly see, and you can do the Raman imaging in 2D sense, and you can find that where are the forms are distributed. So, orange form 3 and blue form 1. 20 like this is Raman imaging. So, you can do a spectroscopy, and same time for the polymorphic term you can do basically imaging as well. So, spectroscopy identify the polymorphic form are present or not, and imaging reveals where they are located in the tablet. So, one can see not only like a form present, but also find out the location by combining Raman spectroscopy along with the imaging. You can monitor the distribution of compound during formulation. So, when API is mixed with excipients, you guarantee that each dose contains the correct amount of medication. It should not happen that in a tablet there is only 70% of API whatever is recommended, not only 60% is there. It has to be 100% with plus minus few percent, that can be 2%, 3%, maximum 5%, but not more than that, otherwise there will be no therapeutic effect. To ensure that the Raman imaging can be done. Ensure even distribution for critical for meeting the quality standard, maintaining the regulatory compliance, and safeguarding, most importantly, safeguarding the patient health by delivering a safe and effective product. That is done by Raman spectroscopy. So, drug development, Raman spectroscopy is used to analyze uniformly the distribution of different compound within the formulation by mapping the spatial distribution of different component in the tablet or in the capsule or whatever in the dosage form, and one can monitor the distribution of compound in real time, monitoring during the formulation process. So, by integrating Raman and inline manufacturing process, it is possible to adjust the formulation process on the fly to ensure uniformity and quality. This is important, on fly to ensure uniformity and quality of the product that a pharma company is making. So, that is what one can do, monitoring the distribution of compound during formulation. So, 8 vitamins tablets were mapped, here are those 8 vitamins, and like a Raman spectra identify using database. So, this is the chemical distribution based on MCR decomposition, this is imaging data one can have. So, here multiple compound are shown in each distinct color, right. One can have a vitamin B12, vitamin B3, citric acid, the calcium phosphate, magnesium oxide and chromium oxide and all those, and each of them are exhibiting different vibrational modes. So, one can identify in each composition what all are present, and then you can have a chemical map of those. Raman makes sure that where they are by doing the imaging. So, spectral match can one of the compound reveals the spatial distribution of each of these component that we see here. So, here you identify molecules and here you look at their distribution in the tablet or capsule whatever we have. Now, you can even characterize the defect or any outlier that is there in the final product. So, you can characterize the defect and outlier in the final product. This is important for ensuring the quality, safety, efficacy of the medication. How you do that? You identify this defect by compromising the product from reaching the patient. So, if something is wrong, much before it released, you can detect it and stop it, right. So, you can detect outliers and manufacturers can maintain the consistency across the batch. This is extremely important, quality control, quality assurance is very important. So, and that helps you in adhering the regulatory guidelines, uphold the integrity of their brand. You see, once name is gone, everything is gone. So, brand value is extremely important, and that Raman makes sure that brand is intact, and therefore, Raman is used in manufacturing post marketing surveillance to analyze the surface of the final product, identify if there is a crack, inclusion, improper crystallization, contamination, anything should not happen. One can analyze and identify the outliers. You can have the outliers to prevent future occurrence. You ensures the consistency among the batch, right, by comparing the spectra of the product. You investigate the cause of defect and analyze the molecular structure and composition of this region. So, that is what one can do, and here is one of the examples. So, look at this is the tablet, and non-compliance of a pharmaceutical tablet because of presence of contaminants. You can find it out. Here is a spectrum in the dark spot, here is that, and this is our API. So, visual inspection if you do, you do not probably see it, but the visual inspection flagged a non-compliant of the target with a dark spot, here is the dark spot, and chemical identification match the defect spectrum to a charcoal, not an API. Charcoal was mixed here, and you can find it out here, right. So, quality control insights demonstrate how Raman analysis can be rapidly identify the foreign material or contaminant present in the pharmaceutical product. So, this molecule, if the API was present, a spectra should have been like this, but charcoal was there, you can look at it. Now, Raman imaging can provide non-destructive level free method for ensuring the product compliance, and Raman role in pharmaceutical quality assurance for not only detecting and identifying the chemical moiety, but also looking at their distribution is very important, right. So, what is the key takeaway from the pharmaceutical industry is highly regulated. You cannot compromise on the quality, right. Stringent documentation required to ensure the safety and efficacy of a product, and Raman appears to be an important tool in this landscape, offers a non-destructive analysis. It provides the detailed insights into their chemical composition of the molecular structure. Also the imaging can do, so like distribution can be done. It can identify the raw material of active pharmaceutical ingredient, also the finished product, and that ensures it is meeting the rigorous standards by regulatory requirement, right. So, both quality control, production setting, Raman can be used as a key tool. It provides the immediate and accurate data on the chemical property of material by identifying the impurity, if something is not there properly added to the tablet. So, this ensures the batch product is consistent and safe for use, and it supports the compliance with regulatory requirement. You can combine with 21 CFR part 11, which is mandate of now regulatory requirement. It secures the traceable electronic record. You can just record the data, keep it there, and everyone whosoever wants to see the data can do it. So, that aligns with the regulation. Raman enhances the quality of your product, enhances that all data is intact, reproducible, traceable, and it is ready for audit, right. So, it supports the compliance with pharmacopoeial standards, USP or EP standard of the other regulatory framework. So, that is all for today, and let us continue our discussion on Raman spectroscopy in the next class. Thank you very much. Look forward to see you in the next class.